Everyone. I want to make a quick video describing what I believe is not being really clarified to the public and to the scientific community regarding the mRNA technology of the COVID vaccines or chemical solutions. Now, the concept here is that the mRNA code calls for RNA polymerase to translate the strip into a functional protein, and the functional protein is the spike protein, which supposedly is found on the coronavirus SARS-CoV-2 on its nuclear envelope. So on the protein capsid, you'll have these spike proteins called S proteins. These proteins, their role is to bind to the ACE2 receptor proteins along human cells. Now, the ACE2 receptor proteins, I'll just show you quickly here, is the angiotensin converting enzyme 2. And these are located along many different cell types in the human body, in lungs, arteries, and heart. They lowers blood pressure by catalyzing the hydrolysis of angiotensin 2 into angiotensin. Now, the SARS-CoV-2 spike protein attaches to the ACE2 receptor protein of the human cell. Now, in order for spike proteins to attach to receptors or protein receptors, they have to have high specificity, and also it must be energetically favorable for these proteins to attach, and there has to be optimal binding between the protein and the receptor. So the spike protein had, in order for the virus to actually enter the human body, it has to mimic what is actually going on in the human body. So human cells have ACE2 receptor proteins because at some point there is going to be exosomes which have these spike proteins from the human body being released under stress, being released under chemical stress, physical, the presence of different neurotransmitters or stimuli. And those exosomes will have spike proteins on them that will attach to the ACE2 receptor protein and release their contents, their nuclear contents, into neighboring cells, causing a chain reaction, causing for either cell destruction or causing other things to happen in the body, such as, if you look at this case, there's a catalyzing the hydrolysis of angiotensin 2 into angiotensin, but in this case, it lowers the blood pressure. So we look at the spike proteins. They are supposedly present in the human body, or else the ACE2 receptor protein would have no use. Okay, so having the ACE2 receptor proteins in the lung tissue, for example, if there's exosomes present due to chemical stress or stimuli, changes in the human body, they're going to release exosomes with these spike proteins. These spike proteins will attach to neighboring lung tissue with the ACE2 receptor protein. So in the COVID-19 vaccines, what the pharmaceutical companies are doing here is injecting you with the mRNA code surrounded by a lipopolysaccharide nanotechnology, nanohydrogel, to facilitate the distribution of the mRNA into the cell, into every cell of your lung and throughout the body. To make what? To make the exact same spike protein. And the concept is that the spike proteins are then going to be released through exocytosis from the cells, and then that would cause then an antibiotic immuno response, causing the antibodies to be developed countering these spike proteins. So then it'll remember, be able to provide some sort of immunity or some sort of protection when the real virus comes in contact with the lung tissue. My question is that these spike proteins that are being coded by the mRNA, if they're already naturally being produced by the body under certain chemical stimuli or physical stress, chemical stress, if these spike proteins are already present in the body, and they have been studied extensively, if you go to literature, a simple search in Google Scholar, you can see even many vaccines have been developed using the ACE2 receptor protein. Why are we allowing a foreign substance made in a laboratory to produce a spike protein that can naturally be produced in the body and through chemical stimuli and physical stress, chemical stress? If we know that there's ACE2 receptor proteins around the body, around the tissue, and these spike proteins supposedly have to be present to attach to these receptors, why are we looking at injecting mRNA code, a very, in my opinion, very novel and without substantial safety data on their effectiveness and also the health impacts later down the pipeline, 6 months, 1 year to 5 years to 10 years from now? And we're allowing then the host cell to be the machinery driving the translation of the mRNA into the spike protein, the spike protein which supposedly has strong affinity for the ACE2 receptor protein, and the ACE2 receptor protein therefore is naturally found throughout the body, throughout the lung tissue. Therefore the body must be able to make its own spike protein through its exosomes and be able to develop that. Another question is, if we're allowing the cell to release these spike proteins, supposedly, why isn't this causing a cytokine storm? Why is the novel virus causing the cytokine storm as opposed to the spike protein? If in fact what causes the response to occur is the presence of the spike protein within the bloodstream or around surrounding tissue, if that's causing the immune response, then why wouldn't the mRNA technology injection cause a cytokine storm where a person can enter into cardiac arrest or enter into severe reaction in the lungs, or even seizures, or leading on to death? We don't know. Like, if we look at people that are dying from COVID-19 with the lack of ability to breathe, they're ending up with fluid in the lungs. There's a cytokine reaction. Now, when we look at that situation, basically we're injecting through this technology foreign mRNA fragments and allowing them to make spike proteins within the cell, which would naturally be produced in the cells of the lungs, because the cell is going to make spike proteins through natural means and release them through exosomes to attach to neighboring tissue. So my question is, are these shots aimed at making spike proteins when in fact the spike proteins are present within the human body? Why aren't we stimulating the body to release these spike proteins naturally, or through exosomes, or the release of exosomal tissue, which has been studied extensively for at least the last 15 to 20 years? That's my question about this, that in fact if there's spike proteins being generated by the own cell machinery, then why are we injecting humans with mRNA technology where in fact mRNA fragments, if there's any mistake in the coding, it could cause some problems? If a foreign protein is manufactured, and if it's not an exact novel protein within the cell will recognize this, and it could cause apoptosis, it could cause some intracellular reactions within the cell that could be damaging. The proteins could cause other issues within the cell. So we look at this case, I don't see that the spike protein is the novel need in this case for anyone to develop any sort of immunity against a foreign agent, which is using the exact same human spike protein which is needed to attach to the angiotensin converting enzyme receptor protein. So that's why when we look at the cases and causes of or the source of the SARS-CoV-2, people that are in the bandwagon that it is made in a lab in Wuhan or in a foreign lab, is that the spike protein is so specific for the ACE2 receptor protein, it must have been derived from humans and added as a vector in the template, in its RNA template. Well, in fact, if it's a spike protein so specific to the human cell, then the human body must be making the spike protein within its tissue and lung tissue or other parts of the body. So this spike protein, which is naturally then created by the body through exosomal tissue in response to external stimuli, how can we release more of that? Now, it doesn't make sense then if the antibodies are formed based on the spike protein configuration. Then if the spike protein configuration it matches exactly what the body is releasing, then would it make any difference if the spike receptor protein had a few differences in amino acids or not? So the whole concept now of a virus, is it real, an actual real entity, or are we confusing this with exosomal tissue and being released as a response to external stimuli, whether it be physical, chemical, or radiation? Now, that is my main question, is now we're injecting foreign substances with very controversial, in my opinion, ingredients, including aborted fetal tissue, PEG, nanotechnology. We're injecting that in the human body to make a spike protein which the body would naturally make under external stimuli. And this spike protein is highly specific for the ACE2 receptor proteins surface proteins found on neighboring cells. So if it has high specificity for the receptor protein and is energetically favorable for bond attachment, then why not stimulate the body to release more of this specific spike protein? And if it's very specific for the ACE2 receptor, then how can we have many variants of this supposed virus? If the many variants, what else do they have in difference than what we would see in the novel SARS-CoV-2 when we look at the different variants? Are they different in the receptor protein? In the spike protein? Is there small differences there? If it were that the case, then are we increasing the bond attachment between that and the ACE2 receptor protein? If it's off by a few amino acids, it may not make a difference. But if it's off by a lot, then there would make a huge difference in the attachment. They call it bond attachment between the receptor protein and the spike protein. In that case, there would be no effect of the virus on the body if it cannot bind tightly to the ACE2 receptor protein. And if the ACE2 receptor protein is highly specific, then the variants, either the variants are matching what you would see from a novel SARS-CoV-2, or the variants are not really inexistent and they're being confused with exosomal tissue from the natural human body. Now, this is what I see, is that we're injecting foreign ingredients made in a laboratory to enter every cell of your body to make a specific protein called the spike protein, which the body would naturally make through exosomal tissue to attach to neighboring ACE2 receptor proteins. Why aren't we stimulating the body to make more spike proteins naturally through physical means or chemical means, if that's the case? Or are we confusing SARS-CoV-2 with exosomal tissue? And this is the theory that I've been sticking with since the beginning of this pandemic, because of many reasons I'm not going to go over in this video, but I believe firmly that there is no need for foreign mRNA tissue to be injected artificially and by force through nanotechnology to every cell to make spike receptor proteins that will be released through exosomes to stimulate an antibiotic response, when in fact the body itself makes its own spike proteins through exosomal tissue under external stimuli, which are highly specific for the ACE2 receptor protein. So the ACE2 receptor protein is like a lock and key concept, right? So the induced fit concept between a protein, receptor protein, and an attachment or spike protein in this case, and the receptor. When we look at depending on the specificity, now we know the ACE2 is very specific. It can have some variation in the amino acids, but after a certain number, it's going to be very weakly bonded and has no effect in entering the cell. So if we look at when they say variants, I don't believe that the variants would have to make such a huge difference in the invasiveness of the virus, and also causing such a much more immunoresponse than the normal SARS-CoV-2. The reason is that if the bond is very strong between the ACE2 and the spike, that's what's needed for there to be an actual connection. And there shouldn't be any other extra response involved here. But we're dealing with something that should be naturally found in the body. If there's ACE2 receptor proteins all around lung tissue in the human body, then there is for sure spike proteins being released by neighboring tissue at some point. So the concept here of artificially inducing the production of spike proteins within the cell is unnecessary and not really called for, unless we're looking at a further agenda that could be involved here, and then speculation. But in my opinion, this is a highly risky situation, dealing with a concept that really doesn't make sense anymore. We want to make spike proteins artificially from external injection, and to attach to neighboring ACE2 receptor proteins, when in fact the body makes spike proteins through exosomal tissue naturally, and this is part of the natural human response. So I'm not too clear about that. I need to do a little bit more investigation, but there must be, if they've studied how to make spike receptor proteins as an mRNA template, then the scientists must have studied how spike proteins are released in the body, how they're being promoted through exosomal tissue and fragments, and how they attach to neighboring ACE2 receptor proteins. Okay, anyways, guys, thank you very much. Take care. Bye-bye.